Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm very excited to be here today with Dr. Andrea Ganna, who's Associate Professor at the Institute for Molecular Medicine Finland at the University of Helsinki, and also a Research Associate at Massachusetts General Hospital and Harvard Medical School. This is actually Andrea's second episode on the podcast. Deep in the middle of the pandemic, we talked about COVID-19 host genetics and some of the really interesting work that him and his group and collaborators really around the world were doing there. We're back today to talk about, I think, a couple of really interesting concepts. So how genetic data can provide a lens into proteomics, into clinical trials, and into the health system more broadly, but also a lot has changed since then with respect to AI. So Andrea's doing some really interesting work, building foundation models, looking at health system data. So we've got a lot to cover today. Andrea, thanks for taking the time to join me.
Thank you, Patrick, for having me back.
One thing that's always impressed me by you and your work is that you manage to cover a lot of ground with your research. You do, complex trait genetics. You're often on the frontier in organizing some really interesting collaborative science. So for people who aren't familiar with your work, can you give a little bit of a sense of what you're working on today and also some of the themes you're focused on in the future?
Yeah, sure. So I think in the past 5 years or so, we have focused a lot on the use of genetically large-scale biobanks and the intersection with epidemiology, clinical trials, and in particular, focusing on polygenic score, and they can be used to study disease progression, how they can be used to make trial more efficient, and other creative application of polygenic score. Now, in the past year or so, that sort of get maybe a little bit less excited about the polygenic scores and focus more on the use of other source of data that now we have really at scale, which is electronic health record. And here in Finland, we are building a large databases of electronic health records, some of them with genetics, some without. And so I'm combining these two areas and transitioning more into the AI application on electronic health records.
You had a recent paper where you showed polygenic scores as well as electronic health record derived scores across a number of different biobanks. And I think in that paper you showed that they were probably largely complementary, right? And also there was cross comparability across biobanks. Is that underpinning the reason a little bit why you started to get more interested in electronic health records and less so in polygenic risk scores? Or maybe break that down a little bit for me of why that shift now?
I'm always really interested in the electronic health record. I had this sort of going to genetics because genetics is sort of the source of information, omics information that is more available at population scale. So it's kind of coming back to the origin to a certain extent. I think that, when you think about polygenic score and the information electronic health record, you're right, they're complementary. And actually, this is a really nice property of genetics. This is complementary on the other type of information. And this is probably the only complementary omics that we have discovered in the past 30 years or so, right? We rediscover every year new omics. We have now proteomics being the big things, but they fundamentally, all these omics capture some smoking, alcohol, and other environmental factors, why genetics truly orthogonal. However, genetics, it has some limitation when we think about polygenic score. It works very well for disease prediction, which means for screening, right? If we set aside the use for biological discovery, which is really, of course, very interesting. And that is, this is an area that we think a lot how to implement it. And the UK has been ahead on that, but it's very difficult. It's very difficult to change any screening programs. And then when we think instead about genetic use in a clinical setting, which is about treatment response and progression, there is where we see that maybe the value is not so large as maybe hoped.
Yeah, interesting. Can you talk a little bit about using polygenic risk scores in trials? Because it gets at all three of these, but it really gets at progression and treatment response. What have you learned through your work on how polygenic risk scores could be used or should be used in trials, and where is there maybe not as much promise? Because it's been in the water for a little while. Can we use them to accelerate time to event or other sorts of approaches in clinical trials? So I'm interested to get your take on that.
Yeah, so I think it's important first to clarify two concepts which polygenic score can be useful in trial. One is the prognostic enrichment. So it basically identifies people that have a higher risk of the outcome of the trial. And the other is the predictive enrichment, which is finding people where the treatment works better. Now, for the prognostic enrichment, of course, they work. And now, big asterisk there, I will come back. But sort of they work if the outcome of the trial is cardiovascular diseases. We have polygenic score for cardiovascular diseases. However, really what the sort of pharma company are really interested in, where the real key point is there, because there are so many things that have prognostic enrichment, right? Cholesterol and so on, is the predictive enrichment. So to find group of the population where the drug works better. And there is where the polygenic score maybe they sort of have not shown that yet very clearly. They've shown it, and here just one step back, they've shown it when the outcome of the trial is well predicted by the polygenic score. So if you think about statin, the outcome of a statin trial is cardiovascular disease. We have fantastic for cardiovascular diseases. So like already in 2015, I think Meg et al. Has shown that when you take trial, the polygenic score can stratify the effectiveness of statins. So there is good. But then when we think about, we did an exercise some time ago with different pharma to bring together their trials or information about the trials where they also have genetics. But when you look at the outcome of most trials, they are very, very specific, right? They are not looking at simple things for where you have a polygenic score. You're looking for biomarkers decline, composite endpoint. And for those, we have not developed good polygenic score. And so by definition, we cannot do a great job in that sense. I think this is the realization I arrived after thinking about the problem for a while.
Yeah, interesting. So do you think then maybe you can provide an example of where if the trial endpoint had been included in a big biobank like UK Biobank, for example, and you could construct the score, do you think that in theory they could be useful there? But the problem is we don't measure the same things in population biobanks that we do in trials. Is that the core of the issue?
I think that's the core of the issue, and it relates to the issue that we have not done a lot of GWAS of disease progression. We have done a lot of GWAS of case control. And so because trials are about disease progression, we should have better genetic marker for those. However, we can do a lot of trial of disease progression. We have biobanks, but there are two problems. One is how you define progression. Everyone has slightly different ideas. The second is that progression is probably less heritable by definition, because people have already been in contact with the healthcare system. There's so many variables, the age when you get in contact. Right. And so there is a big question mark even if we will have a consistent definition of progression, if there are really so much signal to find there. And last point is that we do not have, in many instances, good predictor of treatment response, even outside genetics, right? If age and sex something somewhere, there is a nice paper on the Lancet where they did the predictor of treatment response, where they use different variables. But the point is that if in a drug like, GLP-1 to say you don't see large effect of age, sex, and BMI, probably, you will not see something magically popping up by a polygenic score, right?
Yeah, that's really interesting. So what is useful for that? Is there information hidden in the electronic health records that would be a better proxy for the predictive versus the prognostic that you laid out, or are we missing good biomarkers in general to help see the distinction between those two?
So There's probably something, right? If you think about cancer, they are based on biomarker, the trial itself. So, they have a treatment response that it's very strong based on the mutation that you have. So I would imagine biologically there's something there. And then, of course, we don't forget pharmacogenomics, like the classical variant. Also, HLA has an effect in a sort of treatment response. But I think so. Some people say that theoretically we have those information in the electronic health records, comorbidity, such demographic characteristics that have to do with treatment response. And the reason why we don't see it in the trial is because the trials are too small. But then if you take very large databases, like this Lancet publication I just mentioned, you can start to build a predictor of treatment response or predictor of which drug would be better for you, given you have type 2 diabetes, for example. You can choose between different drugs. So I think there is some people that believe that there is opportunity in the EHR, but then by just doing post-analysis, post-doc analysis of trial, you don't see this heterogeneous effect.
Yeah, interesting. You mentioned that you're shifting some of your research agenda into focusing on foundation models for electronic health records. There's a clear analogy to the boom that we've seen in large language models. It is a lot of it is language, right? Structured, semi-structured, and unstructured notes. Then we also have disease ontologies and medical terminology ontologies. Can you give a little bit of a sense of what's the state of the art today when it comes to applying AI to large-scale medical data, and where do you think the big opportunities are to push that forward?
Yeah, so I think first you need the medical data, and that's, I think, what at least here in Finland we've been working a lot to put together. There are a lot of methods developed on mimic, publicly available data set, but you really need large-scale, high-quality data, and so the harmonization it's extremely important. So it depends. I think the big conceptual shift that this model can do, and they can go from predicting, a binary outcome, disease to not disease, to predict a sequence of events that happen in your future medical history. So theoretically, you can build, 2,000 logistic regression random tree forest and obtain more or less the same thing. And probably you will do the same, if not better. But it's just the conceptual shift, right? Now you can do it all at the same time. And this one, you can do it just on the data. Then there is the second revolution coming in, which is you don't just have your data, you have the entire medical knowledge on your hands that come from the LLMs. So you can imagine to have a language model nested inside your observational data that give knowledge to the model building itself. It gives that sort of counterfactual knowledge that maybe is missing in just the observational data. One typical example is that if you use observational data and you sort of remove someone from study, this person's cardiovascular risk will decrease instead of increasing because, there is a bias by indication in observational data. But we know from trial that statins should lower the cardiovascular disease risk. So that knowledge is there. It's just about taking it and put it inside.
Wait, can you explain the point of why when you remove them in observational, you see the opposite effect?
Well, because statins are prescribed on people with high cardiovascular risk, right? Okay. So the model learned that statin equal is given to people with high cardiovascular risk. So removing statin, it just, it just have that, it doesn't have the right counterfactual.
It doesn't get the causality wrong.
It gets the causality wrong. It gets the causality wrong. Because it's not randomly prescribed.
Yes.
So one way is to put that knowledge there, and that can be done through, for example, mandibular randomization, other method. I mean, genetics is fantastic to build the causality there. But you can also imagine to have some tools at the exit of the sequence of the model and then work on the sequence and say, okay, this makes sense, this does not make sense. Let's refine the output itself.
So I want to just use a very simple analogy and we can see where it breaks down or not. But what the large language models do is next token prediction, right? And we can think of that as, in a way, what we're trying to do in the health system, where you want to know for a person at a particular moment in time. I think you said earlier, what's the next series of events that is most likely, or probability distribution over the next most likely set of events to happen to them. Is that the right way to think about it? There's a lot more questions you can layer on top of that. And with large language models, we also have very sophisticated evals on lots of different areas of knowledge. You can think about applying the same sort of thought process to say it's really important for us to understand, the response to particular treatments. We're going to spend a lot of time really juicing the algorithm in that particular area. Is that how you think about it? We basically have this huge search space and different ways we can apply this, or is there another analogy that's more helpful to wrap our minds around it?
No, I think it's exactly what you described, with the caveat that it's not only about predicting the next token, but predicting when the next token happen. So there are two dimensions, the dimension of time, the dimension of prediction, and with the fact that we are really bad in predicting diseases, for example. So the sort of the probability distribution over these different tokens that you can generate, cancer, cardiovascular diseases, hypertension, it's all very, very low. And so the model gets very confused. But I think the disease prediction is one angle, which is interesting. But again, it's like polygenic score. Why do we care about predicting the disease onset? I don't know, to change our screening program, but then we never change them. So I don't know. But the other is about other things, like a drug sequence. For example, it's quite easy. I mean, if you have been taking statins from the pharmacy every 3 months, pretty sure you're going to define a future sequence of when you keep taking the statins, right? So you can start to predict other healthcare utilization, which is not simply a disease and not disease. And for planning of healthcare systems, it is, I think, more powerful maybe than just doing disease prediction.
What is the big constraint today, even at FIMM, where you've got a ton of well-structured healthcare data? I imagine data quality and quantity is probably still a constraint. How much is on the data side? How much is algorithm development? If you could break down a little bit where the bottlenecks are and where you expect them to be.
I think it's something we are learning day by day. The biggest obstacle is always the legislation to get the data. I think at the scale we are thinking, there is a nice data minimization principle in the GDPR that doesn't really apply to these type of things. So I think here in Finland, we're being quite forward-looking that, but there are many other places where it's very difficult. In UK, I mean, the idea has been there. There's been the foresight model, for example, in trying on NHS or other things. It's very difficult to put it legally. And then the data itself, you need to clean it, harmonize it. And that's a huge task. And I think here, FIMM has done a fantastic job, because there were fundings to do something else, which is the GWAS and drug and genetic discovery, but no funding agency pay for you to do a big QC and data harmonization. So this is a learning lesson you need to learn from other things. And then they need to stay in secure computing environment with certain limitations, disconnected from the internet. So how do you get the LLM inside? How do you get a lot of GPU working and so on? And that's where we are now. I think, let's say we train everything, then the next big problem that we're already thinking about is how do you export this model, right? They contain sensitive data. And so that's sort of the next, I think, challenge to think about.
What do you have any thoughts on how you would tackle that? Because there's already concerns about distilling the weights from low language models. So I suspect the same concern will be there around distilling information from medical models.
Yeah, it's an open question. We've been working on some differential privacy solution. We've been trying to convince the regulator that maybe if we do it carefully, we don't put it on GitHub, but we do it through, secure transfer. It's feasible. We still don't know. We're still studying that problem.
And in terms of the algorithm itself, or the set of algorithms, how do you think about what that is? You have, let's assume that you've got access to data. It's clean as it can be, and you're starting to actually design the foundation model algorithm that you're going to run. Can you because we hear these stories about Anthropic and OpenAI spending tens of $billions on these monster runs. There are also smaller companies that do, much more lightweight, more lightweight ways of training these deep learning models. Can you talk a little bit about how you think that we should think about building foundation models for medical data?
So I think we are now using Mamba as an architecture because the transformers were too computationally expensive. And I'm not a machine learning expert, so this is what
I'm Googling Mamba in the background. So
people in my group are more expert with. But with this new architecture that we have, which basically doesn't go to back, it doesn't need to leverage the entirety of the sequence, because that's another difference with language model, right? What's really mattering in health is what's happened the last 2 years, right? What's happened like 10 years ago is not necessarily so important. And with this model, I think we have maybe 3-4 billion tokens. So basically events in the life of around 7 million people. With this architecture and this size, we can train it on, few GPUs. So nothing that, nothing impossible compared to, large language model. So I think it's still feasible.
Yeah, it's really interesting. How much do you think this is going to be useful for some of the more, basic science discovery? Because you mentioned this before, but genome-wide association studies and other genetic studies, they have medical applications, but they also just teach us something very fundamental about biology. When you think about organizing and interpreting medical data, you've done work on proteomics and polygenic scores and lots of other kinds of omics. How do you think these things are going to fit together to teach us something more fundamental about biology as well?
And you mean like the integration with large language model, for example, to I've seen fantastic application popping up even here at FIMM, where you basically have language model that go and take the knowledge extracted from all these different omic studies and bring it together. I think the way we are able to combine knowledge across studies now with this language model has become incredible. So I'm definitely positive around that. Foundation model apply to other omics. I think it makes sense as much as you can understand some causality out of it. And that's the most difficult part. Because while in healthcare prediction has still a meaning, I don't think that, predicting a protein means, having a matrix of protein, just remove some and to predict a cell in your protein by individual matrix does really matter, right? You do all these things because you want to understand the counterfactual, what's happening if I will change that protein. And there, I think it's where, in gene expression, you see all these perturbation experiments that they're doing. But in other omics, for example, I think about metabolomics and proteomics in human, we do not have large perturbation experiments, right? So how do we, even if we build a foundation model on these omics, we will learn prediction, but we will not learn causality. And that's probably the challenge that we need to face there.
Yeah. What of all your work for the last 5 years or so has surprised you the most? I'm thinking about the thing that you've worked on where you had an intuition going in, and then you just completely flipped 180 based on what you expected going in. Can you think of something that's taught you the most or surprised you in some way?
Well, we've done some work recently in removing genetic from proteins, with the idea that if you remove genetic, so you have a genetic effect on a protein, so a cis-pQTL, but also trans-pQTL, and you remove that genetic effect, so you basically residualize, so you retain a protein that is genetically adjusted. You were testing the idea that these proteins would be a better predictor of the disease. And my expectation is that these things, just doing naively, these things will not work because, in many cases, you will remove good genetics, right? because some of this genetic is also associated with the diseases. So removing the genetic of the protein is not good. But actually it turns out that most of the protein disease pair, they get better when you remove the genetic. They don't get worse, the meaning that there are very few cases where the actual protein is causal to the disease. And it's really a reflection that most of the proteome is not causal to disease. It's just that or confounded by, the usual things, smoking environment, or a consequence of the disease, that manifestation. And so even if you do this naive approach without thinking what is causal and what is not, you actually tend to improve your signals for protein disease pair. And this is consistent with some recent work by Alicia Martin and Alde that has shown that, there's a lot of non-causal effect of proteins in diseases, a lot of environmental work and also work by others that have looked into this space.
Yeah, that's interesting. How do you, there's a lot of work going on in proteomic risk scores. How does this insight propagate through to that? Because it still carries a lot of signal, right, about disease state, but it can be really difficult to pull it apart from the impact of a therapy, the impact of environment. All of those things are going to wash through into the protein space, where obviously they don't into genetic space, right? How does that change how you think about proteomic risk scores?
I mean, I think it makes, so yes, the environment is the main reason why protein predict future disease events. And in fact, we show that if you remove the genetic of this protein and you redo a multi-protein score adjusted by genetics, so genetically adjusted multi-protein score, you do a better prediction of diseases. Yeah. So this is an example in which genetics, it's actually covering the effect of the association between this protein with the environment, which is the real driver of most of these diseases. So the way to resurface the importance on the environment that was a little bit masked by genetics.
Yeah, that is super interesting. Tell me about working in genetics and medical records in Finland more generally. What are people who don't have access to the data or aren't particularly familiar with FinGen? What are some of the big advantages that you have in the system, and what do you miss? What do you need international collaboration for? Because you've run a lot of international collaborations and you have, I think, affiliation across the Atlantic. So I want to hear a little bit about what Finland has that's really unique, but then also where you need international collaboration to do your work.
So yeah, Finland has good laws, which it turns out to be kind of important in these settings. So we had the biobank law back in the days, and now the secondary health data law, which is then being used is the base for the European Health Data Space Act, which had been approved by the Parliament this January. So these laws has opened up the possibility of reusing secondary health data and biobank samples for carrying out this study. So it was really the legislation that made this possible. The second has been this sort of a big support on the use of health data and the use of AI for health data. We have a new Ellis Institute in Finland that is really shows as a sort of moonshot to work on health data. So I think that this is sort of the ecosystem of health data and genetics has been always centered in the sort of country strategy. We still struggle. I mean, these are the sort of European approach. We are all under the research area. We're not like in US, you have some nice biobank, I think about Penn, but also NGH. They have good biobank that are nested in the healthcare set system. So when you want to recontact the patient, when you want to return some information, when you want to run an experiment or a trial, it is much easier than what we can do in Finland. Or something like UK Biobank, or also our Future Health, right? Because you still need to send a letter to people that to be on there to open, and then they need to be invited to a certain center there. You're not inside the healthcare system. So I think that is probably where the translation of some of these findings is complex. We are now using genetic plus AI to predict people that we think that will have abnormal creatinine, cholesterol, and so on. And we're struggling, right? We send them a letter, but then where do they go? How do we get the data back? there is like still a wall between the research endeavor and the clinical world that I think we still need to find a way to take down.
Do you have any thoughts on how we could better break that wall down?
Yeah, I think fundamentally you need to run a research study nested in the healthcare, right? The biobanks should be sort of naturally created by the, if you think about electronic health records, it's not that we have electronic health record because we wanted to do research. We have electronic health record because they are generated by the healthcare and we are smartly utilizing. So we need to generate that omics, that genetic as part of the healthcare and then reutilize that. And it doesn't mean sometimes it will not be very useful or cost effective. And that's where the sort of governmental money or founder money should come in to support the generation of data to enable research and understanding. But I think it's better delivered inside the healthcare system than outside.
Yeah, I want to go into detail in one of your recent papers, which is the one where you did basically, I think you called trial emulation using genetics. You applied that method to, and a reason I want to segue to this is because I think it is a good example of one of these things you could imagine running in a interesting way linked into a healthcare system or set of healthcare systems where you're running trials. Could you just summarize the paper for people who haven't come across it? What did you learn there, and what were your takeaways from it, especially beyond it was for cardiometabolic trials? So what can we take away for trials more broadly?
Yeah, so basically we are doing trial emulation. Trial emulation, for those that are not aware, it's simply trying to emulate randomization using observational data. And you do that by using inclusion-exclusion criteria of the trial and then using matching. And we have done that in biobanks. And the value of doing the biobanks is that we have also genetics. So we started to think what can we do with genetics when we do trial emulation. The first things we learn is that even if you have 500,000 people, trial emulation are extremely needy for sample size. Because when you do this matching, you really throw away people, right? You say, I want for this specific patient that takes the drug, another one that takes a comparator. It needs to have the same characteristics. So if you see, we start with 500,000, but we go down in our trial emulation to sample size that are smaller than the original trial. But that's the first learning lesson. The second is that what do we do with genetics? And there are some idea here. The first one is that basically genetic works as a sort of quality control check if your trial emulation is going well. You can build polygenic score for everything, right? Even for things that you never measure in your observational data that are measurable in a subset. And then you can say, oh, is my polygenic score well aligned in the two arm of my trial emulation? So it works as a sort of orthogonal evidence. But is just a check, right?
Yeah. So you don't select on it. You use it to see if you've unintentionally biased one arm or the other. Yeah, that's really smart.
That's the first one. The second that comes naturally, say, well, but why then, let's say you find that, a polygenic score of a certain biomarker is unbalanced between the two groups, why you do not adjust for that? And we show that, no, don't do that, because it's actually, first, polygenic score are not causal, so you might adjust also for things that you shouldn't adjust, but also because they're very, very poor predictor normally of the biomarkers, so it doesn't help you so much to adjust for that. And the third point is that, so as a next step, we suggested how actually to do it properly, which is not to use polygenic score, but is to use a sort of Mendelian randomization. But it's a Mendelian randomization where the genetic instrument is on the confounder, is not on the exposure. Typically, in Mendelian randomization, you will use something to know if an exposure is causal, to use genetic as an instrument to know if an exposure is causal to an outcome. Here we asking we use it to detect confounders in the trial. And so we can do this screen and then arrive to say, okay, look, you have all these possible confounders that we think are true confounders, and you should adjust for those. If you have measured them, good, adjust for that. If you have not measured, too bad, just be aware that that is a confounder in your data, and you need to sort of be careful about the interpretation.
Yeah, that's really interesting. I was going to ask if you think that this trial emulation approach will be an effective way to do real world evidence post trial, but it sounds like you'd need a ton of data to do this, right? You would really need to, because you mentioned that I'm putting in quotes here, the emulated trials, the sample size were often smaller than the original trial, and that's okay, but you need a ton of data to actually use this to systematically look at the impact of these therapies in a phase 4 kind of retrospective trial. Would that be accurate?
Yes, so I think emulation trial, it's a really fascinating methodology, and I think it would be used more and more to support, trial result, to generalize trial result outside that. I don't know if genetics is going to be, the thing that is needed. I mean, genetics is a fun additional methodological tool, but this trial would be run more. It is, though, the key point of a trial emulation, the most important thing of all is to have a good comparator. So to have a semi-placebo drug that is not the drug you're interested in, but is something else. And that's difficult. It doesn't apply in every scenario, especially when there is a clear, first-line medication, second-line medication, third-line medication. And I think in a country like UK and Finland, where there is a national health care, there are a lot of guideline and cost effectiveness. We cannot do great trial emulation because we don't leave the doctor the freedom to, choose things too well. So I think, someone told me once that we should introduce some randomness in doctor decision making. That when they have the option between two drugs, that should be randomized. They shouldn't be then deciding. So that will allow to run better trial emulation in the observational data.
I'm gonna go on a limb and say that person was probably not a doctor. They're probably a statistician.
No, yeah, exactly.
If people want to follow your work, you're pretty active on Twitter. You've got a website. Are you hiring? Are you looking for collaborators? What's the most useful thing for you right now?
I'm not active on Twitter anymore. I sometimes post things on LinkedIn. I don't know. Follow me.
They can find you. They know where to find you. Yeah.
I just maybe mentioning that, and you were one student of the Lena Penton School of Human Genetics. So we have also this year as the second year we are starting after COVID. So that's a great opportunity for last year PhD student to really interact with very good human genetic faculty. So that's a way to meet me and many other people. So I think the application are until March 6 or 9. So there's still some time.
Oh yeah, great. That is a great shout out. It is, and if people miss it this year, then next year it is hopefully that it becomes an annual thing again. But yeah, Lena Peltonen School is it's pretty special. Did you do it when you were a student, or did you join as a faculty member later?
No, as a student.
You did.
Yes. Yeah.
Cool. Yeah. So for those who aren't familiar, it's basically, I think it's a 1-to-1 ratio of PhD students and PIs. Both industry and both academic and a little bit of industry representation as well. But what I really liked about it is everybody presents over the couple of days, the students, the PIs. So you get a little bit of a flavor of what's happening on the frontier of human genetics and it's a small group. It's probably 40 people, something like that. Is that about right? Yeah. So yeah, highly encourage it if that's your thing. Well, thank you. I really appreciate you taking the time to join us today.
Thank you, Patrick.
Great. And thanks as always, everybody, for listening, and we'll see you next time. Thanks as always for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sanogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sanogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy-Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.